Okay, okay. So we were talking about hypothermia as an anti-cancer treatment using temperature that exceed the physiological optimal level, which is 37 degrees Celsius, our body temperature. And we are using the temperature of 40 to 43 degrees Celsius. But why 40 to 43 degrees Celsius? Why not 40 to 50? Why not 38 to 50? Okay. The reason why it is set at 40, because there was a research elsewhere that stated that at the temperature of 39.6 degrees and above, cancer cells died rapidly. There was a Japanese study who established the temperature to be 39.6 degrees Celsius. At that temperature, cancer cells died rapidly. But why 43? Why not higher? Why not 45? Why not 50? Basically, the 43 degrees Celsius is set to prevent the dying of our good body cells. All our cells are made of protein, and protein will be denatured. In other words, the protein which is in our cells, our good cells, will also be killed if the temperature heating them to more than 43 degrees Celsius. Okay, so that is the reason why for the mild hypothermia, it is set to 40 to 43 degrees Celsius, because when this mild hypothermia is actually administered, both the cancer cells as well as the good cells are exposed to this temperature. While we want to kill the cancer cells, we want to protect the good cells from being damaged as well. So that is the reason why setting it at 40 to 43 degrees Celsius. Okay, so let us do some research on hypothermia now. First, we look at the hypothermia research developments. When did hypothermia research started? What about hypothermia in recent years? Okay, has they become more popular? Okay, so we head to the PubMed. Okay, in case you don't know, this is the National Library of Medicine in USA. Okay, it's known as PubMed over here, PUBMED. So the address is pubmed.gov. Okay, so it is a database containing all the research articles on the field of medicine. Okay, and so it is just like a Google, okay, a search engine. You just go there and type in the keywords. So for example, I type in hypothermia cancer, and you will be able to come out with a search results like this, 18,407 results. And the articles and the research articles will be actually arranged chronologically, no, not chronologically, in a particular pattern, okay, listed over here. And from there, you would be able to read at least the abstracts. Some of the articles, for example, if they are free, okay, if they are free, not paid, then it is actually listed as free PMC article, and you can read and download those articles. For the others, they are not free, then in that case, they will usually be able to give you an abstract. Okay, abstract meaning that a very short summary, so that you know what this research is all about, basically, and most likely they also give you the results as well as the outcome. So in case you want to read the full article, if it is a paid one, you will have to see the publisher of that particular journal, and then perhaps purchase. Or if you are a member, for example, of the university in Tel Aviv, then in that case, you can go through the library and actually download the article from there as well. Okay, so this is how you do the research over here. So 18,407. And over here, it also gives you a map over here. Okay, give you a diagram showing when was the first article retrieved from the database, when was the latest, and they give you a pattern. So you can see from here, there is an interest level. So in this particular case, hyperthermia, cancer, these two keywords gave us more than 18,000 articles. The first one appeared more than 60 years ago, almost 70 years, in 1946. And you can see from there that from this diagram, okay, that you can see that at first, the interest on hyperthermia was very, very low until at this stage here, it started to actually pick up. This is probably, I think, 1/3, probably in the 1960s or 1970s. It started to pick up and then it dipped again. But if you look at the recent years now, the number of articles appearing in the database has also increased already. Okay, so this is how we do the research. So let us do a little bit more. So you can key in the keywords again. Now you add in another word called treatment, for example. Again, not much different. Okay, not much different. The pattern is about the same. So, but we can continue this line of research. For example, if I want to interested in finding out how hyperthermia, what kind of hyperthermia research has been done on prostate cancer, I type in hyperthermia prostate cancer, and you can see that 501 results appear here. And again, if you look at the interest level in the number of articles, it started only in 1977. Okay, the first article. Okay, so basically much later as compared to the other research here, but still 40 over years. And you can see that the interest seems to be fluctuating. But in the last couple of years, it's quite a lot. Although, of course, 2023 is still not over yet. And that's the reason why the bar is still very short over there. So you can see from here, combined hyperthermia and radiotherapy for prostate cancer, a systemic review. Again, this is a free article. Okay, and this is pretty new, 2022. Okay, so these are the research. Again, if you look at breast cancer, also plenty of articles, 1,400, and started earlier, 1954, but recently plenty of articles in this particular area here. Again, free article, you can see that review conventional against advanced therapeutic strategies. Okay, it involves hyperthermia inside there. So you look at ovarian cancer, you got it there as well. Okay, so the pattern is about the same. The trend in recent years using this method of hyperthermia has gained more momentum. Okay, pancreatic cancer, okay, you can see that also increase in this area in the recent years. Liver cancer, also the same results. So some types of cancer draw more interest than the other type. Okay, stomach cancer, you can see that also 500 over results, but the pattern is up and down over here. So basically, colon cancer, you can see that. Okay, all, of course, I did not go through all types of cancer. I'm sure if you have spent time looking into the database, you would probably be able to find out all the other cancer that have not listed, have not done the research over here. You probably can find some research articles on those type of cancer, such as thyroid cancer, such as what other cancer? Brain cancer, brain tumor as well. Okay, so I just want to summarize it for you. Earlier on, we just look at hyperthermia and cancer. We also briefly look at hyperthermia, breast cancer, for example, colon cancer. And I just want to do a quick observation here, summary for you. Okay, so research actually started 1940s. So when people ask you, have you got any proof that hyperthermia is good for treating cancer? Yes, you got it there. Okay, you can confidently tell them that treating cancer using some form of hyperthermia started actually in the 1940s. It's already more than 60 years old. But the number of studies on hyperthermia in cancer has increased steadily over the years. Okay, and more studies are being done to investigate the use of hyperthermia on specific types of cancer. Okay, the different types of cancer in recent years. Okay, so that is one area that I want you to know, the trend, the history of it. Next, we look at briefly the different types of hyperthermia. Again, those of you who have been to our first webinar sometime last year, I would have introduced this in detail, but today I look at it from another angle now. Okay, hyperthermia, basically you want to achieve the effect of hyperthermia only on a local specific location. So for example, one side of the breast or the prostate. Okay, then the second type of hyperthermia is usually termed regional hyperthermia. Okay, for example, you know, in a section of body abdomen, let's say the stomach, also plus the intestine. So it's a region, not on a specific location, but a bigger area zone. And the last type of hyperthermia that had been used in the cancer treatment is what we call whole body hyperthermia. Okay, as the name suggests, it is performed on the whole body. So visually, this is how it looked like. Okay, local hyperthermia. Local, the word local means at a specific location. Okay, so these are with an applicator. Okay, for example, in this case here, most likely this is on the lung cancer, on one side of the lung. So in the second case here, the lady with the applicator, the hyperthermia applicator is focusing, most likely is in the small intestine, large intestine, or perhaps in the, in the ovarian cancer, or in the region over there. So that is local hyperthermia. Regional hyperthermia is a bigger area covered. Okay, for example, in this case here, you can see. And lastly, whole body hyperthermia. The whole body is undergoing hyperthermic treatment, except the head, of course. Okay, the whole body is exposed to the extreme temperature of around 40 degrees Celsius or slightly higher, depending on the researcher. And except the head is outside here. Okay, the head cannot be too hot. You want to protect the head. So, but you want to heat up the whole body. Okay, so these are the three general types of hyperthermia that have been used in cancer treatment. So let us look at the interest in this hyperthermia type over the years. Which one is more popular now? Which one was more popular last time? Okay, so has a specific type of hyperthermia become more popular in recent years? We don't know. Let us look at the research. Again, we go to the National Library of Medicine and we just key in some keywords. Local hyperthermia cancer. It gave us almost 20,000 returns. And you can see from there, in fact, the local hyperthermia started again, just like the original research we have done, 1946. And again, the trend is a lot more in recent years. Okay, and again, you will look at the research article in detail, local hyperthermia in head and neck cancer. The mechanism, they discuss the mechanism of this hyperthermia, the application, and of course, all the advanced features. Okay, and let us look at regional hyperthermia. Regional hyperthermia in this case here, less number of research articles on this, 3005, and it started later, 1963 to today. But again, the trend is the same. Okay, the trend is the same. More and more research in the recent years. Whole body hyperthermia, of course, among the three types, this is the least popular. Okay, and you look at the trend. Look at the trend of application. It started only in 1970, then became very, very popular. But in recent years, it seems to have died down. Okay, the interest in this hyperthermia, this type of hyperthermia has become less popular in recent years. But still, there are many, they are still able to find 501 articles on this. And there are some publications until today. So let us summarize what we have just seen over here. Local hyperthermia, 46, 23, recent years a lot popular. Regional started 1963, and then whole body started in 70. So in terms of the number of papers, you can see from there, almost 20,000, 3,005, 500. So the observation is among the three types of hyperthermia, local hyperthermia seems to have the longest history. Local hyperthermia seems to be the most popular type of hyperthermia in recent years. You can see from here. Out of the 20,000, you can see very high, the concentration in this area here as compared to the other two types. Whereas whole body hyperthermia started last, of course, became very popular, gained the momentum, but seems to have lost its popularism in recent years. Okay, so that is a trend. So what does it tell us? In other words, in recent years, local hyperthermia is becoming more and more popular. If you dig out any articles on hyperthermia, on cancer, the chances of this particular article, this particular study on local hyperthermia, it is much higher than the other two types of hyperthermia. So let us look at the interest of these researchers. Okay, these researchers are primarily researchers from university. They can also be from the hospitals. Okay, they are practitioners. Some are pure researchers, some are practitioners. Now, the first type of interest of using hyperthermia is hyperthermia is applied as a radiosensitizer or a chemosensitizer in combination with radiotherapy or chemotherapy for the treatment of many different types of tumor. So the word sensitizer, of course, comes from the word sensitive. basically they are using hyperthermia to make the radiotherapy more sensitive or make the chemotherapy more sensitive. So that is a meaning. So a lot of research, a lot of interest is actually on using hyperthermia. In other words, they want to find out how to make the radiotherapy more sensitive and make the chemotherapy more sensitive. In a short while, I'll explain to you why, if radiotherapy is effective, why do they need a radiosensitizer and what is the advantage of using a radiosensitizer? Likewise for chemotherapy. So why did so many people fail chemotherapy? How can they improve chemotherapy? They are looking for chemosensitizer. And incidentally, hyperthermia has been used as a chemosensitizer. So this is one area of research. The other region of research is how to improve the effectiveness of hyperthermia delivery in the clinic. What else can be enhanced? Is it the temperature? Is it the duration? Or what else have to be added in order to make hyperthermia more effective? And of course, the third type of research more in recent years is on the technical features of hyperthermia. Okay, to better control and more effective of home of heat delivery. And previously, the type of heat delivery was primarily using infrared, but nowadays, in addition to infrared, starting with infrared, they also use radio frequency. Okay, radio frequency. They also use circulating water, a hot circulating water. And in more recently, they also use magnetic hyperthermia. I will show you a little bit of an understanding of magnetic hyperthermia. Okay, in what is commonly named as thermal ablation. Okay, where they inject certain magnetic substances into the tumor itself and use magnet to induce externally to react with the thermal substance in order to cause the tumor tissues to get heated up to a much higher temperature. Okay, we shall take a look, a glimpse of it later on. But let us do the research now, chemotherapy and hyperthermia. Okay, again, plenty of interest in this area, especially in the recent years. Okay, likewise for radiotherapy, less, but still you can see that in recent years, the number of articles written on this subject here has increased tremendously. Okay, so similarly, on very specific type of cancer using radiotherapy, in this case here, prostate cancer, using radiotherapy and hyperthermia, there were also quite a lot of articles on this subject here. Okay, breast cancer is the same. Okay, so liver is also the same. I quickly go through some of these since I have already did the research before this. Yep. So therefore, a lot is in the use of in the use of hyperthermia as a radiosensitizer or a chemosensitizer. So let us focus now on why the researcher want to use hyperthermia as a radiosensitizer. So use of hyperthermia as a radiosensitizer is really to make radiotherapy more effective for treating many different types of cancer. Okay, so hyperthermia, of course, is used in combination. It is just, I mean, this is one way of using it. So why does radiotherapy need a radiosensitizer? Then we had to understand how radiotherapy works. Okay, and what are the limitations? Of course, radiotherapy works on solid tumors. And it says over here, solid tumors are thought to contain a substantial population of cells which are hypoxic. Hypoxic comes from the word hypoxia, means low oxygen or lack of oxygen. Because much of the cells kill, okay, much of the cell kill caused by the ionizing radiation is the result of oxygen-dependent free radical mediated attack on the DNA. Whereas the hypoxic tumor cells are known to be radio resistant and are therefore a barrier to cure. Now, what does it mean? Simply means it's like this. For a tumor, we know that the tumor cells started in a region of low oxygen, where there is very poor blood circulation. If an organ or if a section of a body has poor blood circulation, it is likely to become cancerous because that is the region of low oxygenation. Very low oxygen, in other words, it is hypoxia, hypoxic in nature. And hypoxic tissues are prone, okay, to cancer because cancers, okay, are anaerobic in nature. They multiply without the use of oxygen. Whereas our normal cells are aerobic in nature. They love oxygen, but cancer cells love low oxygen. So low oxygen in this particular case, for the radiotherapy to work, they need the oxygen-dependent free radical mediated attack on the DNA. In other words, they need plenty of oxygen, okay, so that they can initiate the radical attack on the DNA of the tumor cells. But unfortunately, the tumor cells, many of them or most of them have very, very low oxygen level. So what can we do then in this case? So in that case, although you want to actually kill the tumor cells, but unfortunately, they are what we call a radio resistant because they are very little oxygen in these tumor cells. So you cannot really bring about the free radical attack on the DNA of the tumor cells. And therefore, it is a barrier. And of course, so how do the radiologists work on the cancer patient then? In order to increase the effectiveness of the radiotherapy, the oncologist will have to increase the dosage of the radiotherapy. In other words, use higher dosage, okay, more intense radiotherapy, okay, then only can bring about the attack on the hypoxic tumor cells. But then a higher dosage of the radiotherapy also bring about toxic effects to the good cells as well. So that is the dilemma faced by the radiologists. Okay, they use the radiation to kill the the tumor cells, but then the tumor cells are mostly radio resistant, but they have no choice. In order to kill them, they have to use a very powerful radiotherapy, a higher dose, which I'll look at the dosage later on. But higher dosage meaning that they will bring a lot of toxic effect to the patient, and very often damaging the patient's health, the good cells, until the health is irreversible. So hyperthermia now as a radiosensitizer. So local hyperthermia brings about increased blood circulation to the local tumor site, resulting in increased oxygenation. Those of us who have been using the Onetsu device knows this very well. We bring blood circulation, we increase blood circulation to the area of treatment. So for the local hyperthermia, applicator focus on the particular local tissues. For example, let's say in a prostate cancer, you apply the hyperthermia applicator on the prostate area, increases its temperature, and therefore blood flow to the area will be improved. Once the blood flow to the area is improved, the amount of oxygen in the cell, good cell as well as the tumor cell, will also increase. So once that is increased, with this increase in the oxygen delivery, more free radical mediated attack on the DNA can take place. And therefore, it is more effective for the radiotherapy to be on the solid tumor. So that is the reason why hyperthermia is the radiosensitizer. It's used as the radiosensitizer for radiotherapy here. So what does it mean? As a result of this effectiveness increase, the radiotherapy dosage now can be reduced. In other words, without increasing the dosage of the radiotherapy, you can bring about the effectiveness of the radiotherapy already. In other words, the side effects experienced by the cancer patient will also be less harm by the ionizing radiation. So let us look at some papers now. Some papers, I mean, those of you who want to read articles, and you can actually search the database subsequently. In case you want those articles that I retrieve, I can send them to Miss Zoha, and subsequently she can also distribute the articles to you. Okay, so these articles over here, I just take out a small section on each paper to discuss the key feature so that you understand how it is used as a radiosensitizer. So it is using mild hyperthermia. Mild hyperthermia meaning that it is around 40, 41, 42 at most. Okay, as a localized radiosensitizer for deep-seated tumor. It is not on the surface, but it's deep inside the body. Okay, and in this particular case, they are investigating it based on prostate cancer. And of course, they started with mice. Okay, so the method of the study, in this study here, they got 40 male mice. Okay, they injected it with prostate cancer, this particular type, prostate cancer cells, PC3, and the tumor volume were allowed to actually slowly grow, okay, until the tumor size in the male mice has grown to about 150 to 250 millimeter cube. In other words, they injected the prostate cancer cells into the mice, the healthy mice, and then allow the cancer tissues to grow until this size. And then these 40 mice were randomly divided into 4 groups. First group is the control group. In other words, you don't do anything. Second group, using hyperthermia. Okay, in other words, you go and heat up. The third group is performing radiotherapy. Okay, as usual. The last one is using hyperthermia with radiotherapy. Okay, so with these 4 groups, they intend to see what happened to the prostate tumor cells in these mice. Eventually, so this is what they use in group 2 and group 4. Group 2 and group 4 is where hyperthermia is actually used. In group 2 and group 4, RF induced. RF means radio frequency. Radio frequency induced hyperthermia was administered to achieve a target temperature of 41 degrees Celsius in the prostate. Radiotherapy was administered approximately 30 minutes following the hyperthermia. In other words, they performed the hyperthermia on the prostate area for 30 minutes first. After that, radiation was applied to the prostate area as well as in a single fraction. Now, what is this 12 GY? 12 GY meaning that GY is a short form for gray, GRAY. Okay, the proper unit is gray. It is actually a unit of the ionizing radiation dose. Okay, so typically people going for the cancer patient undergoing the radiotherapy, the doctors will have tell, okay, what is the dosage? Is it 70? Is it 80? Or what else? So in this case here, they make use of a very mild dosage of 12 GY, 12 gray here, okay, in a single fraction. One animal then was actually, in each group was randomized, actually take it out on day 10 or day 11, okay, after treatment with this method of analysis. These are just methods of analysis. I mean, these are some chemical tests they want to carry it out, okay. So these are enzymes involved in the apoptotic pathway, meaning that apoptosis meaning that is the committing suicide of the cancer cells. Okay, apoptotic, one way of active dying of the cancer cells. So they go and actually find out how much of the caspase 9 and caspase 3 enzymes are being released by each of the groups of mice on day 11 or day 12, day 10. And the results. Okay, so when they did the experiment, the inoculation success was 89%. In other words, out of the 40 mice, 89% was actually successfully grown to the required size. Okay, I mean, some did not survive, I suppose, but almost 90% of them did. And the average tumor size when they started the experiment, okay, was approximately 189 mmQ. Okay, and it took about 16 days, okay, from the day of inoculation of the injection till the end. I mean, on the 16 days later, the tumor cells has grown to the correct size they want, and on this day they started the experiment. Okay, so following the administration of hyperthermia and radiotherapy. Of course, hyperthermia first. Remember just now we mentioned that hyperthermia first, radiotherapy for the fourth group. where they actually use the hyperthermia plus the radiotherapy. So the mean tumor doubling time, okay, the mean tumor doubling time, this is again another measure. In other words, it takes how long for the tumors in the body to grow, to double its size. For the control group was 4.2 days. Every 4.2 days, the tumor in the body double itself. In the hyperthermia group was 4.5. In the radiotherapy group, it took them 30.4 days. Whereas lastly, in the hyperthermia plus radiotherapy, it took 33.4 days for the tumor cells to double itself. And through the calculation, they find it to have a significant difference between the normal volume of the RT alone, in other words, the radiotherapy alone, and also the hyperthermia plus the radiotherapy group. Okay, so this is another observation apart from the doubling tumor time, also in terms of the volume of the tumor itself. The increased cap space 3 expression was seen in the combination treatment group compared to the other 3 treatment groups. Okay, in other words, in the combined group over here, they also find a lot of these apoptotic enzyme being released in this particular group. In other words, in this particular group here, more of the tumor cells actually are committing suicide. Okay, that's what they meant. So the conclusion of the researchers here, this is what they said. It is still early result, but this early result demonstrated that the successful rate of external malhyperthermia as a localized radiosensitizer for deep-seated tumor is successful. In other words, it is possible to use this method of local malhyperthermia as a radiosensitizer, okay, for deep-seated tumor radiotherapy. So that is the conclusion of this paper. Okay, more details you can actually go back to look at the paper again. The second study I want to highlight here, it is also radio RF induced hyperthermia, again as a radiosensitizer, again in this case here prostate cancer. The method of study, okay, they use DU145 cells, which is a type of actually, again, I mean, the scientists themselves has already identified the genes of many cells, okay, and they labeled them differently, okay. Early on, we saw one type of cell being labeled, and here is another one. DU145. Okay, so they are meant to be the prostate cancer cell. They can be grown. They can be cultured in the laboratory, and they purchase from the developer these cancer cells, and they use it for their research again. Okay, so they were actually cultured as a spheroid until they reach this size. Okay, then they subject them to these 4 different groups here. Hyperthermia, 90 degrees, 43 degrees Celsius, 90 minutes. Okay, and second group, hyperthermia plus radiotherapy at a low dose of 2 gray with 15-minute interval. And the third group, hyperthermia plus radiotherapy at a slightly double the dose, 4 gray, also within 15 minutes interval of applying hyperthermia. Okay, and then of course, lastly, radiotherapy at 2 GY as well as radiotherapy alone at 4 GY. Okay, so this was what they want to compare: hyperthermia, radiotherapy alone, and then radiotherapy with hyperthermia at different dosages, and then see what happens. So the tests were conducted to measure the cell viability. Are they viable or not? They also measure the amount of DNA damage. Okay, again, when the DNA damage, certain things are known to be given out. So they measure those. I think we don't have to go through the very detail. We just understand the big picture here. And also, they calculated the percentage of induced cell apoptosis. Again, apoptosis means the cancer cells actively dying. Okay, they want to measure that. These are the 3 indicators they are monitoring. So the hyperthermia, you can see from there. Okay, they induce the hyperthermia, quickly achieve the hyperthermic temperature of 43 degrees Celsius, approximately starting 15 minutes, and they maintain the temperature for about 90 minutes over here. Okay, in the control experiments, the temperature was actually controlled to be 37 degrees, which is the optimal body temperature, of course. Let us look at the results now. Okay, look at the results. And this is the result from this particular study. The viability means the cancer cells viable, the survivability base basically. Look at the control group, more than 95%. Hyperthermia, less now, at 90, maybe 94%. Radiotherapy alone at 2 gray, the viability is around 92, 93. At 4 GY, it is now reduced to maybe 92. Now, hyperthermia plus 2 GY radiotherapy further reduced, less than 90 now. And then when it is combined with radiotherapy at 4 GY, it is almost 86% viability. So of course, from here you can see that hyperthermia plus the radiotherapy at 4 GY is the most effective as compared to the other 5 types. Okay, so this is one indicator. Let's look at the another indicator now. This indicator is the what they call the mean tail movement. Okay, so meaning that the damage to the DNA. Okay, so the higher the percentage, the more damage is done. Control group, very little. Radio frequency hyperthermia has improved, and the improvement is in fact bigger as compared to the radiotherapy alone. Radiotherapy at 2 GY. But radiotherapy at 4 GY is slightly higher, and radiotherapy with 2 GY is higher than previous 4. But you look at the radiotherapy plus hyperthermia, radiotherapy at 4 GY plus hyperthermia, it has the highest measurement here. In other words, the activity, the effectiveness of this treatment in affecting the tumor DNA damage is the most significant for this particular group here. And lastly, we look at the thermal enhancement factor now. Okay, thermal enhancement factor. Again, the 2GY, yes.
Morning break.
Yes. Break, is it?
Yeah. Morning break.
Let me just finish this particular study. Just 2 more slides, and then I will stop for a short break. Yeah, so the dosage is here. You can see that radiotherapy alone is 0.6, but if you actually combine 2GY plus the hyperthermia, it has increased to 0.89. And the thermal enhancement factor, almost 1.5. In other words, it's more effective than the radiotherapy, more than 50%. And if you look at the 4GY dosage, radiotherapy alone is 7.78, but with radiotherapy and hyperthermia in place, it has gone to almost 2. The enhancement factor is 2.5 times as compared to the original. Okay, so this is how it actually make the radiosensitizer. Yeah, and lastly, in terms of the measurement of the percentage of apoptotic cell, the cancer cell dying. Again, you can see from here, 2GY alone, radiotherapy is 17.8. But with 2GY plus hyperthermia, it has increased to almost 33, a factor of 1.85. And with the 4GY, 27 starting, and then end up with 64. Again, more than 2 times the effectiveness as compared to the radiotherapy alone. So that's why it is effective and it is used as a radiosensitizer. And earlier on, I just mentioned the apoptosis there. You can see from here, the apoptosis is described as an active program process of autonomous cellular dismantling that avoids eliciting inflammation. In other words, systematic causing the damage to the cells. Whereas the necrosis is also dying of the cell, but these are more passive, the natural dying. So what we want is to actually effectively cause the cancer cell to undergo apoptosis. And in fact, this is a percentage of the cell you can see from here that in this hyperthermia with radiotherapy at 4 gray, it is the most effective. So therefore, the conclusion from there is that hyperthermia at 2GY and hyperthermia at 4GY both are actually both causes the DNA damage as well as apoptosis of the tumor cells. And therefore, in this particular case here, the conclusion is that a thermally enhanced dose in the DU145 prostate cancer cell with hyperthermia combined with the dosage of the radiotherapy was observed. Okay, and the effectiveness depended on the radiation dose. Okay, both at 2GY and of course at 4GY. So the conclusion by the author, it is actually effective. Okay, it is synergistic treatment in cancer radiotherapy, even in this particular case to provide lower dose of radiation, especially for this prostate cancer treatment. Okay, so in other words, the hyperthermia certainly enhances the radiotherapy, the effectiveness of the radiotherapy in terms of the radiosensitizer. That's what we learned from this research here. Okay, we stop here for a short break.